right, I guess we'll get started. Thanks everyone for coming to this special seminar and for enjoying the Greek food, or if you're live streaming, sorry you're missing the Greek food, it's excellent. But it's my pleasure to welcome Dr. Tina Franklin here to UC San Diego. Dr. Franklin and I met at the Society for Biological Psychiatry meeting where she was presenting a poster on some of the work that she'll talk about today. And I thought her work was very exciting, relevant to a lot of the stuff that we're doing here. And as the organizer of the Psychoneuroimmunology Interest Group, I thought it would be great to have her come and talk about some of the work that she's doing. And luckily, she was in town for the Society for Neuroscience conference and agreed to come and talk with us today about her work. Dr. Franklin got her PhD at Rutgers University and is now a postdoctoral scholar at the Yale School of Medicine. So warm welcome to Dr. Tina Franklin.
Thank you. So the turnout turned out to be much larger than than I expected. So I set this talk up in a way to kind of have an informal discussion. So if you are confused about something that I've said, or if you need me to repeat it, feel free to interrupt me. And we should, yeah, I just want to make sure that as we're going through this discussion, that everybody is on the same page, and then we can actually start engaging into a real conversation. And the reason why I'm bringing this up is because a lot of the work that I'll be presenting today is preliminary or work that I'm currently analyzing, and I've set the data up to be completely transparent. So feel free, please, I'm encouraging, I need feedback at this point. All right, so the title of my talk is Self-Specific Ablation of Microglial Pattern Recognition Receptors, RAGE and TLR4, alter susceptibility to depressive-like behaviors after chronic unpredictable stress. So my work really focuses on how stress can initiate and propagate sterile inflammation. So what I mean by sterile inflammation is inflammation that is completely independent of either microbes or pathogens or any exogenous activator. So what we know is that stress can initiate a lot of these inflammatory signaling pathways through the release of factors that are called danger-associated molecular patterns, or DAMPs. And these are a few examples of the DAMPs that are known to initiate sterile inflammation. So these DAMPs can then signal for different signaling, can have different signaling pathways, and one of them is the inflammasome pathway that is first initiated through the ligation of these DAMPs to what we call pattern recognition receptors. So toll-like receptor, RAGE receptors are examples of pattern recognition receptors that then initiate the activation and translocation of NF-kappa B to the nucleus to then initiate transcription of various inflammatory factors and cytokines that have been shown to promote disease or psychiatric disorders. So what we know is major depressive disorder as a psychiatric disorder is a major burden on North American and US health. And what we can see here is that 64% of patients that have a diagnosis of depression also report severe impairments associated with MDD. So it's been challenging for physicians to treat some of these impairments, given that now we know only 1/3 of patients will be responsive to SSRIs. that's been a major drive for the discovery and for the discovery of rapid-acting antidepressants, and also the discovery of alternative pathways that are associated with depression. So one of these pathways is the inflammatory pathway. So here I'm giving you examples of well-documented evidence of inflammatory disruption in MDD patients or patients with other psychiatric diseases that also report depressive-like symptoms. So this evidence has now given us cause to call depression an inflammatory disease, potentially. And the reason why we feel like this is an extremely important psychiatric disease to study in terms of inflammation is because depression is a recurrent disorder. The initial episode tends to be fairly acute, and this is when we see a third of patients respond well to SSRIs. However, a subpopulation of depressed patients will quickly relapse, and subsequent episodes are much more severe and become much longer lasting and chronic, making this oftentimes a lifelong disorder with severe impairments. So what I study are the microglia, which are the innate immune cells of the brain. They originate from the yolk sac and are distant cousins of peripheral monocytes and macrophages. So these resident microglia are really there to kind of survey the environment and either be active participants in shaping the neurocircuitry during development, or during an episode where you are now forming, for example, new memories, or in terms of an insult in either a trauma, sorry, I'm trying to, okay, either a brain injury or an inflammatory response. These cells can quickly move to the site of infection, of the site of insult, and try to attack either the pathogen or initiate a inflammatory cascade that can promote recovery. So why are these cells so important in depression? Well, the reason why we are so interested in these cells is because once the microglia are populated into the brain, they will remain there for the remainder of the organism's life, meaning that any changes that occur in microglia will potentially have a lifelong effect. And we also know that microglia activation or reactivity has been shown to be significantly increased in psychiatric disorders, especially in depression. And it seems that the level of microglia reactivity is correlative to the symptom severity. So if an individual has suicidal ideation, this individual is more likely to have increased microglia reactivity versus a person that has mild depressive symptoms. And that's been shown to be true in several depression-related brain regions. When we look at live patients, so the data that I just showed you was in postmortem tissue. So if we look at live patients, the TSPO detection, which is often used as an indicator of neuroinflammation. So the symptom severity again correlates with the level of TSPO detection and this seems to be dynamic depending on the individual's state. So during a depressive episode, the level of TSPO is increased versus an individual who is now in a remission state or has had successful response to SSRIs or other antidepressants. So to study this, we used the chronic unpredictable stress model. And this chronic unpredictable stress model has been shown to mimic a lot of the depressive-like behaviors or symptoms that you can associate in a major depressive disorder. So some of these are decrease in cognitive performance, which we test with the novel object recognition test, the sucrose preference test, which is an indication of anhedonia if the animal has reduction in sucrose preference, a social avoidance or isolation, which can be tested with social interaction test, forced swim test, which is a test of motivation or despair, or locomotor activity, which is used to assess the animal's general activity, but also can be an indication of sickness behavior. So we're able to disassociate an animal that is sick versus an animal that is showing depressive-like behaviors. And so to do this, the chronic unpredictable stress model exposes the animals to various stressors that range from mild to strong stressors. And these can include peppermint odor, reverse light cycle, so overnight light on or daylight light off, social isolation, cage crowding, so not allowing the animal to have enough room to explore, strobe lights, shaker, forced swim, and the forced swim has been excluded in all of the tests that I will show you despair behavior in these animals. So this is why I've added an asterisk here. Wet bedding, which is fairly stressful for an animal, and restraint stress. So far, does everybody understand? Okay. So this model is our preferred model of stress-induced depressive-like behaviors, because this seems to really mimic what we experience as humans. So the stressors are ranging from mild to strong. Then none of them are really categorized as traumatic. So this is fairly mild, but it's also a great model to mimic the general population's exposure to stress. So some of our preliminary data here is presented here. And what we did is use the CUS model, or the chronic unpredictable stress model, to determine the level of microglial activation and the factors, the sterile inflammation related factors that are altered after CUS exposure. So here in this timeline, you can see that we've incorporated a subchronic exposure time of 3 days or 7 days. So during that period, the animals are not displaying any depressive-like behaviors. Then we have our 28-day CUS model, which is a time point where all of the animals, or the majority of the animals, will show depressive-like behaviors. And then to study whether or not these changes are dynamic or long-lasting, we also incorporated longer time points up to 42 days, where the animals have now had 28 to 42 days of recovery, quote-unquote recovery. And what we then did is we isolated peripheral PBMC, so peripheral blood mononuclear cells, and microglia from the PFC, and microglia from the hippocampus, to determine changes at the periphery versus changes in different regions that are associated with depression in the CNS. And we only found changes robust changes in microglia, inflammatory factors, and the hippocampus. And these changes were most pronounced in HMGB1 expression. So this is mRNA expression. We can see that after 28 days, there's this robust increase in HMGB1 mRNA in the hippocampal microglia. There's also an increase in RAGE receptor, which is a receptor that recognizes this ligand HMGB1 at 28 days. And no change in TLR4, another receptor that recognizes HMGB1. When we looked at later time points where the animals are no longer exposed to stress and are in this recovery or post-stress period, we can see that HMGB1 changes persist up to 42 days. The changes in RAGE are transient but persist again up to 42 days. And the changes that there are still no changes in TLR4 expression at the mRNA level and hippocampal microglia. So, at least in this model, CUS promotes the upregulation of HMGB1 and RAGE. When we looked at the now translational changes, we're looking here at faxoided microglia cells, and we can see that there's a trend in TLR4 increased expression on hippocampal microglia, but this is not significant, whereas there's this very robust and significant change in the level of surface RAGE expression. So now this is showing us that CUS is promoting both the upregulation of RAGE mRNA and increasing the level of surface RAGE expression. During that time, we also collected basal corticosteroid levels, and we can see that in the timeline where the animals are not showing depressive-like behaviors, so 3 days, 7 days, the basal corticosteroid levels are fairly low. After 28 days, when the animals should be now showing depressive-like behaviors, you can see a robust increase in basal corticosteroid. So that's without any recent stressors. And this is an overnight, so 16 hours post the last stressor. We can now see this robust increase in corticosteroid levels. And this in the post-stress period, at 14 days or 28 days, the basal corticosteroid levels are restored. So none of the changes that we see, none of the molecular changes that we see are due to increased corticosteroid levels, at least in the post-stress period. And the animals also do not show any depressive-like behaviors after being exposed to an acute stressor. So the post-stress period, now 28 days after the last stressor, if we expose them to one acute restrained stress exposure, they have a response that's comparable to naive animals, and they're again not showing any depressive-like behaviors in the sucrose preference test. So there is no change in preference of sucrose solution versus water solution. But if we look at other depressive-like behaviors after repeated exposure to unpredictable stress, we can see now that the animals have an increased vulnerability for depressive-like behaviors. And this is the sucrose preference test. So here, this first panel is showing you that CUS, so chronic unpredictable stress exposure, promotes a decrease in sucrose preference in the stressed animals. Here, so if you're comparing the striped line to this line, these are the same animals that we have here that are now separated into two subcategories, where these animals now have received stress for 7 days, and this is not enough to induce a depressive-like behavior. The post-stress animals that have had now 28 days to recover if we don't expose them to stress, do not show depressive-like behaviors. But if we give them an additional 7 days, so after the post-stress period, if we expose them to another 7 days to reinitiate this depressive-like behavior, the behavior is now restored. And what's most interesting, at least to me, is that these behaviors are not all transient. Some of these behaviors persist. So if we look at the novel object recognition test, here where the animal has to differentiate between an object that it's already seen before or a novel object, and control healthy animals will prefer to explore the novel object, you can see that the CUS animals explore the novel object less compared to the control animals here after 1 hour of exposure. And at 24 hours, they perform even worse. If we give them 28 days to recover in an attempt to have now this normal behavior, we can see that still after 1 hour of exposure to the novel object, the animals are not able to differentiate between the novel object and the familiar object, and they perform much worse after 24 hours of the novel object exposure. And this is very similar to what we actually see at the clinical level. So the many patients that do respond to SSRIs will still report a decrease in cognitive performance. So it may really be, this model may really recapitulate the depressive-like behaviors that we do see in humans. Are we good? Okay. If we then look at the molecular changes after the post-stress period, we can see that RAGE levels, so the striped line, compared to the solid blue line, the post-stress animals have a level of RAGE that is comparable to the naive animals. However, when we re-expose them to stress, we can quickly promote a upregulation and surface RAGE. This increase in pattern recognition receptor expression is also shown when we look at TLR4, but it's not as robust. And when we look at microglia activation, we can see that microglia that are normally ramified become hyper-ramified after CUS exposure. So the cells have now longer, more complex processes. And with our show analysis here, we were able to show that CUS exposure does indeed promote hyperramification of microglia in the hippocampus and also in the prefrontal cortex. And this effect seems to last in the post-stress period, although it's much more subtle. Okay. So all of the data that I've shown you so far seems to indicate that RAGE is important. RAGE is upregulated during CUS. It's also upregulated when we reinitiate this depressive-like behavior or phenotype after subchronic unpredictable stress exposure. So what we thought is, okay, well, maybe RAGE is really mediating some of these effects. So we used these global RAGE null mice to determine whether or not the animals show resilience to CUS. And we were able to show that indeed the global knockout animals seem to have partial resilience. So here they perform better in the sucrose consumption test, which is equivalent to the sucrose preference test. They perform better in the novel object recognition test. So now this is indicative of a partial resilience. So so far we thought, okay, RAGE is important, but RAGE may not be the only receptor that is really mediating this effect. And again, if you may remember in the beginning, I told you that HMGB1 is a ligand that is recognized by both RAGE receptor as well as TLR4. So our thought was maybe this is only a partial resilience because other receptors like TLR4 are compensating for the lack of RAGE. So our hypothesis now was, first, let's look at microglia-specific RAGE deletion. This, again, was a global knockout, so it may be that other cell types are mediating this effect. And related to this hypothesis are other receptors like TLR4 also involved in the resilience to depressive-like behaviors. So our approach was to use a specific tamoxifen-induced Cree recombination, and we gave the animals weeks to have a microglia-specific knockdown of those receptors by just letting the peripheral macrophages and monocytes proliferate and restore their expression of the pattern recognition receptors. So the half-life for monocytes and macrophages are 10 to 14 days. So after 20 to 28 days, you should have a complete turnover, versus the microglia that have very little turnover, and any changes that occur in these microglia cells should be long-lived. So why does it look like this? Okay, so I will just let you know what we're looking at here. So the first panel is body weight. The second is sucrose consumption, which is paired with the, I'll use my mouse, which is paired with the water consumption. The fourth panel is forced swim test. This is novel object recognition test, and here I'm showing you the location of the animals in a heat map. This is locomotor activity, and this is the social interaction test. So first in body weight, this is showing that CUS does promote the decrease in body weight, and this was not prevented in the microglia-specific RAGE knockout animals. When we look at the sucrose consumption test, the wild-type animals show a decrease in sucrose consumption, whereas the microglia-specific RAGE knockout animals have comparable levels of sucrose consumption. And we're still trying to figure out what this means because they have different water consumption, so this is now being retested actually this week. Then this fourth panel is showing you the forced swim test, and animals that show depressive-like behaviors will have an increase in immobility time, and the wild-type animals show an increase in immobility time. The RAGE knockout animals have no resilience, and actually the control animals perform worse than the wild-type control animals. So we're thinking that RAGE expression is actually necessary for to prevent despair-like behaviors in rodents. When we looked at the novel object recognition test, which we saw the greatest effect in the global global RAGE knockouts, we no longer see this protection. So microglia-specific RAGE deletion does not protect against potential cognitive impairment. And when we look at social interaction, so the first panel is wild-type control, this is wild-type stress, this is RAGE knockout control, and then this is RAGE knockout stress. The RAGE knockout animals, regardless of stress exposure, have a reduction in social interaction. So none of what we thought was going to happen happened here. So microglia specific RAGE deletion does not protect against depressive-like behaviors. So the next thing we did is look at TLR4 receptor. So one of the things I discussed was maybe the deletion of one receptor induces a compensatory mechanism to another receptor that recognizes many of the same ligands, and TLR4 is one of them. And so we looked at the same behaviors, looked at body weight, sucrose consumption, forced swim test, novel object recognition, and social interaction test. And we can see here that stress does promote a decrease in body weight. The animals do not show any protection against sucrose consumption test. So there's no, the reduction that you see after CUS is not blocked in the TLR4 knockout animals. When we look at the forced swim test, we don't see a protection against the stress-induced increased immobility time. And the changes that we see in the novel object recognition test, and this is again preliminary, so we need to increase this, seem to trend towards a partial protection against cognitive impairments after CUS exposure. So here there may be a partial protection, but again we need to continue exploring this. And when we look at the social interaction test, there is no protection. So the TLR4 knockout animals perform actually worse than the CUS animals, the wild-type CUS animals. So we tried to knock out both receptors to prevent any potential compensatory effects. And so the changes here that we see in body weight are different than what we were expecting. So the wild-type animals are not showing a reduction in body weight, but when we look at the sucrose preference test, they do show a reduction as sucrose preference, like you would expect in the wild-type animals. This is not protected in the double knockouts, so the RAGE and TLR4 double knockouts. However, when we look at the forced swim test, now we can see a protection. So it seems that the double knockout animals now are protected against despair-like behaviors after CUS. When we look at the novel object recognition test, again, this is something that we need to continue analyzing, but I've added this just to show you the spread, but it seems like indeed that the animals are now partially resilient to stress-induced cognitive impairments. But either RAGE or both receptors are important for normal social interaction, and if we delete either RAGE or if we delete both RAGE and TLR4, now there's a reduction in social interaction regardless of stress exposure. So this is complicated, and it was somewhat disappointing, but then when I was thinking about this, we went back and looked at toll-like receptor 4 and RAGE expression and different CNS cells, and the reason why we really focus on the microglia is because they have the highest expression of toll-like receptors, but many other cell types express RAGE and TLR4, including astrocytes and principal neurons, or in this case, what I was studying was pyramidal neurons. So the next thing that we did is we looked at neuronal specific knockouts of pattern recognition receptors, and these are not conditional knockouts, but we can specifically ablate RAGE and pyramidal neurons or TLR4 and pyramidal neurons, and then explore whether or not these animals are more or less resilient to depressive-like behaviors. And what I'm showing you here, the top panel, the green bars are TLR4 specific knockout animals. The bottom ones are RAGE specific knockouts and pyramidal neurons, like chem kinase 2 expressing neurons. And the toll-like receptor 4 deletion did not protect against novel object recognition test deficits, so there's no protection against stress-induced cognitive impairments. And there's no protection against the forced swim test. No, sorry, this is the opposite. The data that we saw in the forced swim test was inconclusive because the CUS animals were showing a reduction in forced swim tests. So this is, again, being retested. But what I want you to focus on is this, where we see is finally positive data that kind of supports what we saw in the global knockouts. So if we delete RAGE from chem kinase 2 expressing neurons, we can now see a protection against stress-induced cognitive impairments. So these animals are performing much better in the novel object recognition test. And they are also performing much better in the forced swim test. So they have a reduction in immobility time after CUS. And so this is now being completed, and we are, the data that I didn't include here, but that we analyzed last week, shows similar trend. So again, so hopefully next time that you see this data, that we'll have more conclusive results on whether or not pyramidal specific knockout of RAGE receptor protects against depressive-like behaviors. So in conclusion, I've shown you that chronic stress promotes the upregulation and increased expression of surface RAGE expression on microglia cells, that the RAGE knockout mouse, the global RAGE knockout, increases resilience to depressive-like behaviors, that microglia-specific RAGE deletion may prevent stress-induced anhedonia in the sucrose preference or the sucrose consumption test, but it increases vulnerability to despair-like behavior in the forced swim test and social isolation. So we're not sure what this really means at this point. And the antidepressant effects that we observed in the global RAGE knockouts may be driven by neuronal RAGE silencing rather than microglia RAGE silencing. So some of our future direction is to have a extensive microglia morphology analysis to determine whether or not RAGE activation is important for the hyperramification that we observe after CUS, or whether or not it is not either protecting or promoting microglia activation. The next thing that we're doing is a molecular analysis to determine the inflammatory factors and the neuronal atrophy that occurs after CUS. So we wanted to know whether or not toll-like receptor deletion or RAGE deletion is protecting against some of the inflammatory factors, expression that have been reported by many groups in the field. The next thing that we want to do, that we are doing, are microglia-specific HMGB1 deletions, and we're testing for behavioral, so we're doing behavioral analysis to determine risk or resilience to depressive-like behaviors when we block the ligand that initiates either RAGE or TLR4 activation on various cell types. So we are also doing the microglia morphology analysis that I described earlier, and we've also started the molecular analysis with a NIDA proteomic grant that I recently received. and also a transcriptomic analysis in these microglia-specific HMGB1 knockout animals. So at this point, I'd like to thank the people that were involved in this work. So these are some of the Duman lab members that participated in this work. The Ornstein lab that have now helped me explore more cognitive, different cognitive impairment tasks that can really help us differentiate between working memory and episodic memory. The Harold lab for providing us with the RAGE knockout mice, and the Yale lab in Kansas for providing us with the HMGB1 and RAGE conditional knockout mice. And of course, the funding. Okay.
Well, I have two questions. The first one, I need. Yeah. Well, the first one is, I think it's, as you mentioned about, there are a lot of molecules on microglia, and we found, individual knockout has subtle phenotypes. So if you really think the microglia is, I think, play a key role, and probably you can just, do microglial depletion using the CS1R receptor inhibitor.
Yes.
So as I think the report, So you can also, they can repopulate and 1 week later. So that I think may give you an answer whether the microglia is the target you want to focus. The second question is, I think I look at the combination, and I think according to our experience, and I think a lot of people, I think, it's a lot, when you either using the Cree line, and really they really look at the, the Cree effect by itself. I think we use the transgenic Cree and a knocking Cree. Every time we see there is a Cree effect. So that one, when you have a two allele, the genotype you use, I think you need a 4 genotype combination, so you can tell whether there's Cree effect. Yeah.
Okay. For your first question, we've already done that experiment. The results were disappointing. So Plexicon has this drug that you can targets the CSR, targets the receptor that depletes microglia, and so we've done this experiment, and we tested for novel object recognition test and the sucrose preference test, and it did not protect against these tests, but it did prevent long-lasting vulnerability. So when we reinitiate the stressors, so for that subthreshold or subchronic exposure, the animals are now resilient to those. So depletion and repopulation of microglia seems to now prevent long-lasting effects, but it does not prevent that initial, at least in our hands, doesn't prevent that initial depressive-like phenotype. The second thing, I completely agree with you with the Cree effect. We've actually, the data seems to point that there is a Cree effect, at least in the social interaction test, that with or without stress exposure, the animals are showing kind of antisocial behaviors. We've been trying to understand what that means, but one of the things that we have done is quantify the level of knockdown. And this is not something that I've shown you here, but we see that there's a 60% knockdown of the expression of those receptors. So part of what we think is that the Cree effect is not complete. So we're hoping that that 30% remainder, like the 30% that we see, is indicative of a partial Cree effect and not a full Cree effect but this is something that I can't address in the global knockouts.
Oh, actually, my second question is not about the Cre-efficiency. Cre-efficiency itself, that's definitely another issue. You're going to look at that.
Yeah.
What I'm saying is, it's not about the Cre-efficiency. It's the Cre itself. If you're using the transgenes, that's a positional effect on mouse behavior. If you knock it in, we see the knocking in has the mild effect and a positional effect from a transgene, but still has effect. Let's say you have the CX3R1 Cre. When you target it in, actually in one animals, and even I think we use a line which target into the 3UTR. Theoretically, you are not going to affect the genes you target in, but you still see effect. Yeah. So if
what would be
one genes, one allele, you will have haploinsufficiency. I think most Cre knock-in is kind of haploinsufficiency. Loss of one allele. I can't conclude a haploinsufficiency, but that's...
Yes. If you have time after, we should discuss because something that is that I do want to address.
Did you look at the change in RAGE expression in other cells, such as neurons or the microvascular endothelial cells?
So the astrocyte cells we haven't looked at. The neurons, that's what I just showed you was partially driven by RAGE expression that we had quantified in wild-type animals. And we can see that pyramidal cells express fairly high levels of RAGE receptor. But those changes are not, so stress does not promote an increase in RAGE expression in neurons. Not sure if the dynamic expression of RAGE is really important here. It may be that the activation of the receptor, regardless of the receptor availability, is what's important. But did I answer your question? So there's no increase in RAGE expression in neurons that we can see of.
About the endothelial cells?
So we did not explore that. We didn't test that. We didn't quantify that.
Thanks for a very nice talk. You discussed some of the stress effects in terms of vulnerability, resilience, and so vulnerability presumably is this group of animals that's exposed to stress that shows an impairment. As I'm sure you're aware, some labs will talk about vulnerability, resilience in terms of the entire group of stressed animals and which animals that are stressed show an impairment, which animals that are stressed don't show an impairment. Yes. Did you look at any correlations between the stress response in these animals with some of those immune markers?
Yes. So that's what we're doing now. Some of the transcriptomic work and epigenomic work is separating so what we're planning on doing is correlating the way that the animals performed to the levels of changes in inflammatory factors. So these, so everything is, so basically like animal 1 is then correlated to itself, like to its own expression. What we don't see is, for example, for like a repeated social defeat stress, you can clearly see a separation between a vulnerable group and a resilient group, and that usually falls around 30% of the animals are resilient. We don't see that in our model. We always see a few animals that are...
Sorry, based on which behavior?
On the sucrose preference test and the forced swim test.
Okay.
So they clustered together too close to really separate out a population, but there'll always be 1 or 2 animals if we're looking at a N of 12, for example. So what we can do is explore some of these changes in a different stress paradigm, like the social defeat stress or learned helplessness. But in this model, it's very difficult to separate out resilient groups.
So I guess it seems like, I mean, obviously the take-home message is it's complicated.
It's complicated,
right?
yeah.
So it seems like some things are going on in the microglia, some things are going on in the neuron, but when you affect the microglia, it doesn't seem to affect things the way you want. When you affect the neurons, it does seem to affect the things you want, and yet that's not where the expression is. So, so it seems to me that maybe what's going on is the interaction between the microglia and the neurons, right? So do you have any ways of looking at that? Have you thought about doing any kind of like culturing neurons to, in combination with the microglia to kind of see how they're interacting? 'Cause maybe it's just hard to see the full picture if you're just looking at microglia or just looking at neurons.
Yes. So we've thought about that. So our model right now is that we think that the ligands that affect the receptors are coming from the microglia, and that the receptors on the surface of neurons are mediating these effects. So we haven't done this in culture yet, but we were able to do that easily. But one thing that we are doing is using that HMGB1 microglia specific knockout to then determine resilience. And so far, it does seem that HMGB1 blockade induces resilience. How do I know if it's an interaction between microglia and neurons? At this point, I don't. But one thing that we've thought about is doing an ICV infusion in the neuronal specific knockout animals to see if I infuse HMGB1, which is only released by microglia, by the way, in the CNS, then I can, it's not a direct evidence, but we can say that, yes, extracellular DAMPs, which we know only happens, only released in microglia, does seem to work through neuronal specific pattern recognition receptors. But that's something that we need to explore, but we haven't done so far. Yes.
Any other questions? All right, well, thank you so much,
Thank you.
Dr. Franklin, for your great talk.